And you can kind of consider the stomach just to be a tube. Yeah.
So the chloride is going through a leak channel. It's not like a one-way thing. It's
Yes, you can consider it a leak channel. Yes, it's an open ion channel. Yes.
So the inside of the parietal cell is pretty acidic. Then. Right?
Well, it can be. Yeah.
Okay.
Yeah.
Okay.
Yeah, because it's hosting hydrogen ions, chloride ions. Yeah, it can be.
Okay.
But through diffusion, it's moving out of the cell.
Okay. The chloride.
So yeah, these parietal cells do have some, some acid resistance. They have proteins that work in slightly lower pHs and whatnot, but it's not like they're operating at an extremely low pH.
Okay.
And we'll talk about the protections against acid in a minute. Yeah.
So what's the role of the potassium?
So that's just the exchanger here. So,
okay.
so this exchanger, I'm running out of space here. This exchanger brings in chloride to put out a hydrogen.
Okay, so just like chloride and wrote potassium.
Sorry. Taking in a potassium and it's putting out a hydrogen ion that I can't write for some reason. Yeah.
So it's exchange for potassium and hydrogen.
Yeah. Why that is evolution. Why don't we just have a hydrogen ion leak channel? There are some reasons, but this is just how the system is built here. Is there a good reason for it? Not really, but that is how it is built. When I say built, this is how it's evolved, I mean. And the reason we have this leak channel is because if we kept on accumulating potassium inside the cell, then this transporter wouldn't work very well. It'd be harder to do this transport. And so you have to leak that potassium out to help drive that exchange. So why have, yes?
Where does the hydrogen and the chloride bind together to make hydrochloric acid?
It doesn't really bind. That's the thing about a strong acid. When they're in a solution, they disassociate. So you just
solution and then they bind.
No, with water they just disassociate. All this is being facilitated with water, right?
Which is why we're not burning our linings of our stomach.
Yeah, the only time you would ever see them combine together if it was in a crystalline form. But if you put it in any solution, they're going to disassociate immediately. Yep. All right. So why do we have hydrochloric acid in our stomachs? What are they doing? Well, first of all, they're breaking down protein. So they break up protein. And so they're targeting disulfide bonds, hydrogen bonds in the protein. So they're really breaking up the kind of quaternary structure of the protein. They're not breaking it up into amino acids, keep in mind, but they're breaking it up at a larger scale. So that means the quaternary scale. Also, acid is really good for killing bacteria. So this is another form of protecting the body from pathogens, right? Acid, a lot of bacteria that would be in our food probably can't handle low pHs, so this is a good way of destroying them. And which we'll see in a minute, they're used to activate enzymes. And to inactivate amylase. So remember, this amylase in our saliva is digesting starch. So by the time it hits our stomach, interacts with that acid, they denature. So a lot of you did the low pH experiment with amylase and you found that your reaction rate went to basically 0, well, that was on purpose. And so we can deactivate that amylase by the time it hits the stomach. And then to activate other enzymes the stomach puts out, and we'll see that soon. I think we'll actually see the next slide. But we good with this? We're good with the parietal cells? Good with the pathway? There are a lot of different pathways in this chapter, in chapter 21, I think. There's a lot. And they tell you all the different forms of transport. They tell you all the different exchangers and whatnot. The ones I need you to know are the ones that I go over in class. The ones I don't go over class, you don't have to worry about. But the ones like this, the parietal cell, you need to know.
What is the water and the carbon?
Just from the environment. Most cells have water somewhere. They're respiring cells, so they're produce carbon dioxide. So, like that. Just from being a cell, they'll have water and carbon dioxide somewhere. Yeah. All right. So, let's draw the pathway of what happens in a cell. So here is our mucosal layer. Let me draw a huge mucosal layer. I need the space. This is the mucosal layer. And so this is then our lumen. Right, swallow that corner now. Alright. So, we detect our amino acids. And peptides, they are picked up by sensory neurons. Right. And those sensory neurons will then talk to a cell called G cells. G cells are called G cells because they put out a molecule called gastrin. Gastrin talks to a handful of different cells. They talk to, but the main one we're considering here is they talk to parietal cells. Parietal cells make what?
HCl.
Yes. They're putting out acid. And so that acid also then triggers other sensory neurons to then put out, well, these sensory neurons will then turn on these chief cells, which put out an inactive enzyme called pepsinogen. Right here. Pepsinogen is what we call a proenzyme. It's not an enzyme yet. It's kind of like a prequel. It's a proenzyme. So these are inactive enzymes. And in fact, what activates them is the acid. It activates them and then turns into an active enzyme called pepsin. So pepsinogen is activated by acid and turns it into pepsin. And so pepsin is an active enzyme. It breaks down protein. Now, why do we do this funny business of putting out an inactive enzyme and then having acid activate it? Why don't we just have these chief cells keep pepsin inside of them and then output them when they get the signal? Okay, regulation. But you can have those chief cells just store pepsin and put them out when they get the signal to output them, right? They can still get input from this sensory neuron, be like, release a pepsin. And then, but why don't they do that? What's the problem? Yeah.
Is it because you don't want to like digest your own stomach?
Yes, pepsin break up proteins. What are your cells mostly made out of? Proteins, right? So if you kept pepsin inside the cell, then you just digest your cell, right? That wouldn't work, right? These would just start cleaving your organelles, your other proteins. It would be horrible, right? You eat, the cell would eat itself. So what it has to do is it has to keep them in inactive states, and then put them outside of the cell, put them into the lumen, where then they can get activated. And so this is what helps prevent cells from basically auto-digesting themselves, is by putting out an inactive form, which then gets activated inside the lumen, outside of the cell. Eventually what happens is also this, running out of space already, the acidity also activates a D cell. And this D cell creates a signal called somatostatin. Might do this in a different color. Pop. But somatostatin basically then negatively inhibits the whole process. Tells cells, stop doing what you're doing. Tells the G cells stop making gastrin. It stops the chief cells from making pepsinogen or releasing pepsinogen, and it stops the parietal cells from creating acid. And so this is the